Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel Science for Everyone. Today, the video is related to microbiology and it is basically related to virology section. And in today's video, I will discuss that how will you diagnose a viral infection. Okay, and I will introduce different techniques that are used to diagnose viral infection. Most of the microbiologists and students of microbiology, they are well known to the techniques that are used to identify bacteria inside a person, inside a specimen. But if they are asked that how will you identify a virus, so then they are answerless because they are not having sufficient information about it. Okay, so keeping this issue in my mind, I will discuss briefly that how will you diagnose a viral infection. In today's video, I will just show you the techniques, that which types of techniques are used in diagnosing a viral infection. And in the later videos, I will discuss each and every technique in detail. Okay, so in this video, I will just show you, figure out the techniques that are used in diagnosing a viral infection. Okay, so let's start with the lecture. Okay, okay, so this is a animated picture. I have taken it from a book, and it is a very authentic book. And I have made a video over this book, that which book is this, and why is this is very much important. And I'm using this book in my videos. So watch that video. The title of that video is that best book to study medical microbiology. Okay, so it is very much easy book, and it can help you a lot. Okay, so if first of all, the first thing is that how will you diagnose a viral infection. The first thing is that if we look here, we start from here. So sign and symptoms, we will look to the sign and symptoms of the person. Okay, for example, we will observe the manifestation of a typical virus infection. And basically, this is a herpes simplex virus. These are cold sores. Okay, these are cold sores, and mainly they appear on over the mouth and over the lips, and it is caused by a herpes simplex virus type 1. Okay, so as a microbiologist, you should know the sign and symptoms of each and every infection. Okay, so if we look to the person, so we can detect that he's having a viral infection by detecting its signs and symptoms. Okay, so if you see a person having a source on his lips just like this, so you can easily tell that it is a type of viral infection, and it is caused by herpes simplex type 1. Okay, and if you want to get into the more detail of it, so what will you look here? The second step is that you will take the cells from the patient. Okay, you will take the cells from the patient, and you will examine it inside a microscope. Okay, and the first thing is that you will look to the cytopathic effect of that cell, of that virus, that how that virus is killing the cells. Okay, so each and every virus kills the cell in a specific way. Mainly virus kill the cell by rupturing their cell membranes and getting out from it. So it is a type of table. You can check it, that how which virus kill the cell in which type of ways. Okay, so you will take the cells from the person. For example, this is the place the person is infected, so you will just take these cells from the person, and you will examine it inside the microscope. You will not see the virus in it, because inside a compound microscope you cannot see the virus, but you can see uh, the the structure of the cells, that how they are killed. Okay, so from there you can detect that it is a viral infection. Second is that virus antigen can be detected by fluorescent staining. Okay, so fluorescent staining is very much important, and it is an easier way to detect a virus. There are different type of stains that are used to stain viruses and cells. So you will apply those stains, and after that you will detect it inside the microscope, and you will see that either these cells are infected by a virus or not. I will discuss these techniques in more detail in the later videos. In this video, I'm just telling you the techniques of detecting a viral infection. Okay, so this is the second way. The third way is that the electron microscopy. Okay, just wait a minute, I want to scroll it down. Okay, so wait a minute. Okay, so the okay, so the third way is this is the third way. Okay, and it is if you're having an electron microscope, so just take the sample from the patient through a needle, and after that through a syringe, and after that place the specimen inside a electron microscope. And in electron microscope you can easily detect the virus. Okay, because in the electron microscope the resolution and the magnification power is very much higher, and you can easily detect the structure of the virus. Okay, so if it is rotavirus, so it will appear like this. If it is hepatitis B virus, so it will appear like this. Okay, so you can easily check it inside the electron microscope. But mostly we are not having the electron microscope in our labs, so that's why it is a bit complicated thing. The fourth is the serological testing. These tests are mainly used in the laboratories. Okay, serological testing means that we take the blood from the patient. After that we centrifuge it, and we then check the serum of the patient. Okay, the serum is the upper part of the blood. When the blood cells settle down and the white color part, the white fluids come out, it is in the upper portion. So these contains the antibodies that are produced against the virus. Okay, so we can detect the special type of antibodies that are specific for the virus. So here come the ELISA test. Okay, and ELISA, the enzyme linked immunosorbent assay, and other the immunofluorescence assays, these all come in the serological testing. Okay, so we detect the antibodies that are produced against the virus. So if we are having those antibodies inside our serum, so we can easily tell that this patient is affected or infected by a specific virus. So this is the fourth one. The fifth one is the mainly this is also conducting all over the across the laboratories, and this is the PCR technique. Okay, this is the PCR technique. And through PCR, what does we do? We just remove the DNA from that virus, and after that we magnify it, and after magnifying it, we replicate that DNA, and we produce large amount of that specific DNA. After that we add specific probes to that DNA, and after that when these probes get attached to our specific DNA, so they and keep this thing in mind that these probes are specific to that specific type of DNA. Okay, of the virus. So when they get attached to the DNA, so they start lightening. Okay, so we can observe that we are having our specific nucleic acid. Okay, sorry, nucleic this DNA. So this is the fifth way of detecting a viral infection. And the last way is that mostly in rare cases this is done, that we culture the virus. Okay, we produce a cell line, and after that we inject that inoculum into that blood, into that cells. And after that the virus start growing inside that cells, and then we can detect it in the microscope, or we can detect its cytopathic effect, that how it kills the cells. Okay, so but mainly this is done in rare cases, because for a virus a cell is important in order to replicate in order for its replication. So we should take a cell, and that cell will be a cell line, or we can inoculate it inside inside an egg. Okay, an egg, we can inoculate it inside that embryo that is present inside that egg. So these are the 6 ways, okay, that how a viral infection is detected. But keep this thing in mind that this way and this way is mostly done in the laboratories, and this electron microscope and this cell culturing, and these ways are a bit complicated and they are mostly done in rare cases, okay. So keep this thing in mind. Mostly when you are an expert, so you can, oh, wait a minute, if you are an expert, so you can easily detect the viral infection from the sign and symptom. But if you want to get more information about that virus, so then you have to do these or such type of tests, okay. So this was all about that how you will diagnose a viral infection. In the later videos I will try to make detailed videos over these each type of techniques, okay. So stay tuned with my YouTube channel and share this video. And if you have understand this video, so like this video and share it with your friends, okay. Thank you for time. Thank you very much.